So we will be talking about the fourth topic in DNA, which is genetic engineering. Genetic engineering is things like making cauliflower different colors or growing ears on mice, not about turning mice into kiwis. So here's this cartoon. Isn't genetic engineering amazing? 2 years ago, who would have imagined such a thing as a turkipede? It's manipulating DNA. It's building new DNA by modifying old DNA. It's carried out by bioscientists or biotechnologists, splicing new pieces of DNA or genes from one chromosome to another. It's a refinement of traditional breeding practices. We've been doing this all along, we're just doing it a little faster. It's also called Recombinant DNA Technology. This requires 4 things. Plasmids from E. coli or bacteria. So that's a plasmid, that little circular piece of DNA. It requires a gene. So the blue piece is the gene that you're interested in putting in. It requires something to cut it, a restriction enzyme. And it requires some glue or ligase for attaching the DNA together. Why is E. coli used? E. coli can be used to make human hormones because it can contain these circular pieces of DNA called plasmids. E. coli has a short lifespan. It's easy to grow. It's cheap. It is plentiful. It can live in the human gut, and plasmids can be transferred naturally between E. coli bacteria. How do you make recombinant DNA? You have the pieces of DNA that you want to put into your plasmid. You have your plasmid. You add a restriction enzyme to chop up the DNA. You mix it together with a molecule called ligase, and then you have these new plasmids being formed. So restriction enzymes cut, ligase acts as a glue, and you have these new plasmids that are formed called rDNA or recombinant DNA. The desired gene is cut out of the host DNA using restriction enzymes, exposing, in some cases, these sticky ends. So you have ends that match up. AATT matches up with TTAA. That's a restriction enzyme. It's called EcoR1. It cuts at a specific site, GAATCC. The same restriction enzyme is used on both the plasmid as well as the gene from the organism that you want to copy. The desired gene is inserted into the plasmid using a ligase. So here's the cut. Here is it coming together, and a ligase glues it together. The plasmid is inserted back into the E. coli bacteria cell. The E. coli is allowed to reproduce the desired product. The product, for instance insulin, is separated from E. coli, sterilized, bottles, and sold by the pharmaceutical industry. Restriction enzymes cut DNA at specific places. These sites are recognized because they are palindromes. So things that we use in our regular everyday language, pull-up is a palindrome, rotator is a palindrome, race car is a palindrome. It means the same thing backwards and forwards. On the DNA, these recognition sites will look like something like GAATTC, and on the opposite direction, you see GAATTC. This is what the DNA molecule looks like before it's cut. And this is what it looks like after it cuts between the G and the A on both of these molecules. GA cut, GA cut. The sticky ends are exposed and allows the genes to fit into the correct place. Here is a restriction enzyme cut. EcoR1 cuts between the G and the A. The host and recipient DNA must be cut with the same restriction enzyme to expose complementary sticky ends. There are more than one type of restriction enzyme. EcoR1 is not the only one. GAATTC is not the only recognition site, so don't get confused about that. It's just an example. Many different restriction enzymes exist. Most come from bacteria. What are some applications of DNA technology? The first is food production, so recombinant tomatoes. Pharmaceuticals are another. They produce antibodies and proteins. Human hormones and products such as insulin are produced. Interferon, somatotrophin, or human growth hormone. The production of BSTs, also known as bovine somatotrophin, so that is bovine growth hormone. It increases milk production in cows. Gene therapy is also something that they're working on, replacing defective genes with non-defective genes. There in Edmonton at the Alberta Research Park, there's a giant fermenter that is used to make recombinant DNA products. What are some uses? These two mice are the same age. One has been given growth hormone and the other one hasn't. The growth hormone that the mouse was given came from recombinant DNA technology. What are some pros about recombinant DNA technology? You can produce human products without side effects, so things like insulin. You can produce disease-resistant strains of plants for agriculture. You can combine characteristics of two organisms into one organism and hopefully get the best of both worlds. You can also get a very specialized form of selective breeding. Things like jellyfish and rabbits equal glow-in-the-dark bunnies. So there are people who are against recombinant DNA technology as well. So here's an example. What have we done? The scientists are saying it's a killer carrot. It's perfectly safe. What are the cons? Some believe that we are playing God. Eugenics, we might use it to select specific traits in our offspring. It's expensive to do. We're not sure about the long-term effects on other organisms. It leads to the development of resistant viruses and bacteria through plasmid transfers. So we are not quite sure of the